Good morning, everyone. This is Rebecca Bailey, and I will be the moderator for today's webinar. Welcome to the April webinar. I hope that everyone is well and sitting very far apart. Today, we will have two speakers for the webinar. Today's speakers are Drs. Iraj Hosseini and Justin Feigelman. Drs. Hosseini and Feigelman will be taking questions at the end of the webinar, and you can ask them questions by entering your question into the questions pane on your GoToWebinar side panel at any time during the webinar, and we will be addressing all of the questions towards the end of the webinar. Today's speakers are from Genentech, and Dr. Iraj Hosseini joined Genentech in 2015 and is currently a scientist in the pharmacokinetics and pharmacodynamics department. His work focuses on development of quantitative systems pharmacology models and preclinical translational PK/PD characterization of biologics, T-cell dependent biospecifics, and personalized RNA cancer vaccines. More recently, he has worked on gQSPSim, an open source platform to help with standardized QSP model development and application. Iraj has co-authored over 20 peer-reviewed journal articles and conference papers, received the ACOP Abstract Quality Award, and was named a SimBiology Scholar in 2015. Dr. Justin Feigelman is a scientist in the area of preclinical and translational pharmacology, quantitative systems pharmacology at Genentech, where he has been for the past 3 years. Prior to that, he worked as a postdoctoral researcher at ETH Zurich, working on multiple computational projects. He has experience in the development of systems pharmacology models for multiple disease areas, machine learning, statistics, and computational biology. And he has a diverse background in physics, computational biology, and biomathematics. His current interests are primarily in the areas of mathematical modeling and the development of algorithms and software to advance the field of computational biology applied to disease physiology and drug development. With that, I'd like to welcome our speakers today, Iraj and Justin, and welcome to the April webinar.
Thanks so much for the kind introduction, Rebecca, and thanks, Rosa, for giving us the opportunity to present today. We appreciate the audience taking the time to listen to this webinar today during this pandemic, and we hope you are all well and safe. So today, Justin and I will be talking about gQSPSim, which is a SimBiology-based graphic user interface for standardized QSP model development and application. Before I start, I wanted to acknowledge the individuals who have significantly contributed to this project. From Genentech, we have Monica, Vidya, Saroja, and Kapil. And from MathWorks, we have Anita, Sietse, Pax, and Fulden. So we've seen an increased interest essentially in QSP models and the applications of QSP models in drug development, pharmaceutical industry, and biotech industry. And often these models are built using a wide range of softwares, workflows, and methodologies. So our motivation for essentially building this app was to facilitate the reproducibility, transparency, and reuse of these QSP models by essentially providing the platform that can help with the technical workflow of QSP model development. We see that there is a lot of QSP models that have been published, but sometimes we often see that the details are missing, and reproducing the results from those models can be tricky. So here the aim is to provide a platform that will help with the transparency and reproducibility. Sort of like with that, we have specific aims in mind. So we wanted to make sure that we are supporting the technical workflow. So we will essentially have a functionality for calibrating QSP models to data collected across experimental conditions. The other functionality would be to be able to generate virtual populations and virtual cohorts of virtual subjects to explore the parameter uncertainty and biological variability. And then finally be able to use these virtual populations to provide robust model-based predictions for different new treatments as well as the new scenarios that we have. So this is the technical piece of it. And on the other hand, we wanted to make sure that this software works hand in hand with SimBiology. So if you think of it like if you're a modeler and you have two displays in front of you, one display has SimBiology, which you are building the model structure and all the different features of SimBiology. The other display has gQSPSim, which helps you with all these additional features and functionalities. So any model that you have built in SimBiology can be used in gQSPSim, and gQSPSim and SimBiology are supposed to work hand in hand. So in order to achieve the goal of being able to share, we made sure that we can have cross-platform compatible sessions. So if you are using Windows or Mac and you want to share these sessions between your collaborators and other modelers, you can easily do that. The software provides essentially a documentation of your workflow for reproducibility and transparency. Now, QSP models are typically long projects. You spend months and years on these projects, and this will provide a nice documentation of what you have done. It is essentially could be a work history that you have done. So if you want to go back and look at a model from 2 years ago and compare the results with today, you can do that. But also if you have a specific functionality, you want to look at a specific calibration that you did or a specific virtual core that you develop, you can go and look at the details in the session that you have built in gQSPSim. So that provides sort of like a documentation as you build your models and as you develop these in QSP projects. So it's an open source platform, and we want to essentially invite anybody who would like to contribute to these to the development of gQSPSim to contact us, and sort of like we can discuss what we would like to include in gQSPSim. And finally, this provides a platform for non-modelers, your collaborators, other reviewers to review all the analysis that you have done using a user-friendly graphical user interface. So this is the motivation for us to build this platform, and we were hoping that this would essentially facilitate the reproducibility and transparency for QSP models. So for today's agenda, first I have a brief introduction for the workflow of QSP model development, and then I will give an overview of the gQSPSim framework, the building blocks and functionalities. Then I hand it over to Justin to demo the app and showcase some of the features of gQSPSim using a simple TMDD model. So we've used a simple model for today because we know that the audience is familiar with it, so they can focus on the features and functionalities of the app. But if you're interested, you can look at the paper that we've published, and we have provided a QSP case study in the paper as well. Now, before we talk about the app, we need to look at the workflow that we have included in this gQSPSim. So Gatkar et al. Has published a paper talking about the different stages of QSP model development. This is typically an iterative process, but it can be divided to different stages. The first one being essentially identifying project needs and goals. This is when you talk to your collaborators, the clinicians and the researchers on the team to define the questions that you need to answer. Once you have that, then you define the model scope and essentially what are the biological things that you want to include, what are the mechanism of action or the data that you want to include, and what is your confidence in those pieces of the data. Do you need to include additional hypotheses? Once you have settled on those things, then you will represent the biology using mathematical equation. This is when you put the model structure together using SimBiology or other softwares. Once you have done that, then we get to the stages 4, 5, and 6, which is of interest for gQSPSim. This is the piece that becomes technical and it can become repetitive. So you will essentially go through this process for every single project using the codes that you have developed. The first one is to look at the model and essentially come up with a single parameterization of the model that can capture the average behavior of the measurements that you have. This is essentially generating a reference virtual subject and sort of like calibrating the model. This will give you confidence that the model structure that you have put in place is sufficient to capture the dynamics of the measurements that you have from your preclinical or clinical data sets. Once you've done that, the next piece is to look at your data, which you typically have a lot of variability, and sort of like explore the knowledge gaps and uncertainties to be able to generate virtual subjects that capture the range of variability that you have measured. This will give you essentially a virtual cohort of subjects. And finally, once you have your model and a virtual cohort, then you can use that to predict the outcomes for new treatments and new scenarios, and then support experimental and clinical design. For gQSPSim, our focus is on the stages 4, 5, and 6. Of course, this is an iterative process. With the new results, you can essentially go to the different stages as needed and sort of like then produce new behaviors. But stages 4, 5, and 6 are the pieces that are more technical, and you need to have reproducibility and transparency when you look at your results from the model. Now, some of the key definitions that we need to focus on are essentially related to virtual subjects, one of the main building blocks in gQSPSim, and it comes in different flavors. We call a virtual subject essentially a single parameterization of the model that would yield model outputs that could or could not be, essentially may not be within the range of measurements that you have. So it's essentially just a single parameterization of the model that will yield some model outputs. Now, a reference virtual subject is a parameterization of your model that would lead to model outputs that are corresponding to the average behavior of dynamics that you have observed in your measurement. So this is the piece that will give you confidence that the model structure that you have put in place is sufficient to capture the dynamics that you've observed in your data. This doesn't need to be unique, but you need to get at least one to get that confidence. Then you have a virtual cohort, which is a collection of these candidate virtual subjects, and they are essentially alternate parameterization of the model, and they will generate a range of outputs that are within the measurements that you have, so within the lower bound and upper bound of measurements that you have. And finally, a virtual population, which is a set of virtual subjects that are prevalence weighted to reproduce selected statistics from your data. So this could be that you want to match mean and standard deviation in your data set or the distribution, and this provides essentially the scenario that you generate your virtual cohort and you look at the statistics on top of the model outputs, and it would match the statistics that you have observed. So the resources that we have for gQSPSim, we were supposed to do a workshop at QSPC this year, and now this has been canceled and postponed to 2021, so we will have that workshop. We have published this app, the building blocks and functionalities, plus 2 case studies in a journal paper in CPT-PSP, and the app itself is an open source software, so it can be downloaded from GitHub. If you go to MATLAB Central, you can download it. You can search for gQSPSim and then download from GitHub. Now, a little bit about like how gQSPSim and SimBiology are connected. So if you look at the workflow for a modeler that is building these models, so essentially you have, again, one display SimBiology. You build your model structure, doses, variance, parameters, species, all the features that you have in SimBiology. And you can have multiple files and multiple models. Then in gQSPSim, you build tasks that are essentially equivalent of experimental conditions. So you define the specific scenarios that you have. You also add other building blocks, such as parameters and data sets that are required for different functionalities. And finally, with those, you will be able to perform a number of functionalities that we will discuss in the next few slides. And then each of them will generate results that are essentially most of the time different flavors of virtual subjects, and they will be saved back in the session file. But they also you can look at the results. You can generate figures, and you can export the results to do additional analysis. So these two platforms are essentially hand in hand. The idea is that you use it for developing models. You build something in SimBiology. You have all of your data sets and parameters in Excel files, and all gQSPSim does it saves the relationships and the mapping between the different components. It doesn't save the content of your files, so you can change things in SimBiology, save it, and then run it in gQSPSim immediately. So that's essentially a feature of gQSPSim. It is a wrapper for all the M files that you would have otherwise needed to develop. So if it's not clear by now, gQSPSim stands for Genentech QSP Simulator. We have 4 specific functionalities and 6 building blocks. So the first functionality is simulation, which gives you the capability to simulate one or more virtual subjects for the desired scenarios. Optimization or calibration to produce a reference virtual subject. Virtual cohort generation to produce a collection of virtual subjects, and finally virtual population generation to give you prevalence weighted virtual subject to match the statistics that you have observed. The building blocks are tasks, which will be used for all the different functionalities, parameters, which is used for cohort generation and optimization, a data set, which is used mainly for the optimization functionality, acceptance criteria for cohort generation, target statistics for virtual population generation, and finally all these would yield virtual subjects or different flavors of virtual subjects, which will be used for simulation. And we will be discussing in more detail all these building blocks. Okay, so let's look at the first functionality that we have, which is a simulation. So the 2 inputs to a simulation is first task, which is a list of scenarios to be simulated. This is an equivalent of the experimental conditions that you've had. The other input is a virtual subject, which could be one reference virtual subject or many virtual subjects. It could have prevalence weights, or it could be all equally likely. So these are the different flavors that you can use for simulation. Now, how do you define a task? A task is essentially, as I mentioned, a virtual experiment corresponding to the specific real experiment that you have. So if you have a preclinical study with a molecule at different doses, those could be your tasks. If you have a clinical study with different molecules in different populations, those could be your tasks. So it's also how you define your model and how you define your variance in the model. And then that's how you can sort of like come up with the task in gQSPSim. The way to do this is to select the SimBiology project and model. Once you do that, it will populate all the different features from the SimBiology model. Then you can select the variance for this particular simulation. So if you have, so example, cyno variance or human variance. If you have doses that you want to select for a particular simulation, or now if you want to select the species, these are the species that you select from the model, and these are the outputs that will be used for plotting or exporting. Now there are some advanced features to select submodules of the model to be deactivated. So think of it like if you have a model with a tumor compartment and you want to look at the healthy individual data and you want to deactivate the tumor compartment, you can use it here in this module here. And finally, the settings, which would be the output times, the maximum wall clock, and whether you want to run to a steady state or not. Okay, so now that you have defined a task, let's look at a virtual subject building block. So this is essentially the parameter values that you have in the model. So if you think of it, the parameter space, a virtual subject is essentially a single parameterization of your model. It's an Excel file format. The first row defines all the model parameters that you want to change in the SimBiology model. And then each row provides the different values that you have for each parameter. And this defines one virtual subject, then the next virtual subject. You also have an optional column called prevalence weights. If you have provided this, then this is the different prevalence weight that you have for each virtual subject. Then they add up to 1, or if they are 0 or 1, essentially corresponding to valid, invalid, we make sure that it's normalized so that the sum is 1. So this will help when we are calculating the weighted averages and standard deviations and other statistics. With the tasks and virtual subjects, you are essentially able to run simulations for different scenarios. The next piece is optimization. The input optimization is, again, a set of tasks that are corresponding to the different groups of data that you want to capture. A data set, which is the collection of all the data that you have for different scenarios. And finally, a parameter file, which is a list of parameters that you want to estimate. The output of the optimization is a reference virtual subject, which is essentially a single parameterization of the model that would provide the best feed to the data that you have provided. The data set, again, is an Excel format. You have 2 columns, group and ID. The groups are corresponding to the different doses, for example, in your study, and the IDs are different animals or subjects that you have. And then you have a column for the time component, as well as the different output data. In this case, for example, we are looking at PK and PD measured at different time points. A parameter file has, again, a bunch of features. The first one is an include column, which tells you whether you want to estimate something or not, and the name of parameters, the scale that you want to explore these parameters. So if you have a wide range for your parameter values, like in terms of lower bound and upper bound, we suggest that you use log scale. If it's not that large, and you can explore it on a linear scale. And finally, you provide an initial guess for the initiation of your calibration. Once this is done, this would provide a single parameterization that we call reference virtual subject. Now, once you're done with this, you have confidence in your model, you know that the model structure can reproduce the behavior. The next piece is virtual cohort generation. It takes tasks, parameters, and now acceptance criteria. So acceptance criteria is essentially a range of measurements that you have and the set of conditions that you need to satisfy. This would be the lower bound and upper bound of measurements at any given time point for the groups that we need to capture. So, like for example, we are looking at 2 virtual subjects here. The blue virtual subject is within all the measurements that we have for the different outputs, so it's a valid one. Whereas the purple one is outside of the range for 2 of the measurements, so that would be an invalid virtual subject. So for a virtual subject to be valid, it needs to satisfy all these conditions that you have provided in the acceptance criteria file. Again, the format is simple. You have the group numbers, you have the time component, you mention which data point and what is the lower bound and upper point for any given time point that you have done the measurement. So this will provide a set of conditions that needs to be satisfied for a virtual subject to be valid. So once you've done this, the output is a virtual cohort, which is a collection of valid virtual subjects. The next piece is now to take these virtual subjects, take the target statistics, which is coming from our measurements, and generate a virtual population, which is the final flavor of virtual subjects. How do you provide target statistics? Again, you look at the different groups that in your data, and for the different measurements that you have, you can provide the statistics to match. So if you have mean and standard deviation at any given time point, you can provide those, or if you have mean, you can provide those values. And the algorithm essentially optimizes the prevalence weights so that the output of the model matches best with the distributions that you have provided. So now you can see that essentially all that you need to do is work with the SimBiology and provide all the different building blocks in Excel files in order to be able to use gQSPSim. So for today's demo, we're going to focus on a TMDD model. This is a simple TMDD model with 2-compartment PK as well as targets in circulation. The data that we have been provided is the free PK as well as the free target and total target. And the idea is to estimate PK parameters and the KD value. The data that we have is 2 different doses, 3 mg per kg and 0.3 mg per kg. The in vitro affinity, we've been provided with that number, and the duration of the study is 21 days. In terms of the measurements, we have the average of the data and the mean and max of measurements. For the demo, we will create appropriate tasks, simulate the model with default values, we try to estimate the parameter values, generally virtual cohort and virtual population, and essentially we do this as much as possible during the time that we have. So at this point, I want to hand this over to Justin to demo the app.
All right, thank you.
Thank you, Iraj. Let me just change presenters for everybody.
Okay, all right. Well, thank you, Iraj. Thank you, Rebecca. Yes, so let me get started with the demo. I'd like to just show off some of the features that Iraj was mentioning here and kind of walk through the TMDD workflow. So we start off with MATLAB console, and we need to launch the app. So once you download the code, you can just do this directly in the app directory. This will start an instance of the main UI. Okay, so that's up. And now we'll get started by firstly creating a session file. So this drop-down menu here, new, that creates a new session. And we need to start configuring the session and start defining the tasks and appropriate building blocks so that we can do some of these functionalities that we want to do today, like the optimization and cohort generation. So we start by clicking on the left-hand side of the app here, which is our session explorer. It shows the session and all the contents of the session. And each of these different tree nodes corresponds to a different building block or functionality. This very top level one is the session. And on the right side, as soon as you click on something in the tree here, the right side will populate with a summary view, which, if you can see this clearly, shows you the information describing this session. So here the name and the last save time and so on. And if you click on this pencil icon, this allows you to edit the properties of this session. So we'll go ahead and do that in order to get set up here with this TMDD model. So I'll call this TMDD. And the most important property of this session is the root directory, which, speaking to the concept of encapsulation, everything relating to this session, all the data, all the results, and so on, they all live within this master directory called the root directory. And this way, if you want to share your session with somebody else, all you have to do is give them the entire directory, and they can put it wherever they like on the file system, as long as you properly specify the location in this root directory path. Everything works relative to that root directory. So let me go ahead and define that here. I had this already set up for our TMDD model. So this directory contains the data and the files necessary for running the various functionalities. Okay, so I specified the root directory. We also have a objective functions directory, which if we wanted to define custom objective functions for optimization, they would live in this directory. We have a user-defined functions directory, which is if you've written a bunch of M files which do various things, which are obviously custom, and you want those to be available to the app for running the models, you can define them in this directory, and this will be added to the path automatically. We have a few more options. One is to use a parallel toolbox. So if you have the parallel toolbox installed, you can specify which cluster you would like to run simulations on, and this will help speed up simulation, but also other functionalities which benefit from parallelization, like optimization. So I'll leave that off for today because it tends to be a bit slower when you run it in your local machine and due to the overhead for this very small model. But it's useful for the context of larger models, or if you want to push this off to a remote cluster like a server. Okay, we also have a few options regarding auto-saving. So if you'd like to have the UI save the session periodically, that's an option. This way you won't lose your work if it crashes or if MATLAB crashes. And before run means that every time you do something, a simulation or an optimization, it will automatically save for you. So then you always have the latest available in case of a crash. Useful in the case of large models or if it's unstable. Okay, so I will just leave it configured like this, all of those things off. And now that that's been configured, we can go ahead and start defining tasks and remaining building blocks. I'll start off by defining the data that we want to use for the comparison with the models. So we have this data set building block. Now, if you right click in the menu, you're able to add a new item. So I'll do that and select it and edit this. And here I would like to load in the data we have available for optimization and comparison. And so those data have already been defined. We have them in this directory. Here they're called data mean. And as Iraj kind of pointed out, this is an Excel file. It contains information about the groups, which are experimental conditions, IDs in case of multiple subjects, for instance, animals in experiments, time points, and the different data points you have available for each of those different conditions and time points. So in this case, we have the free PK, the target concentration, free, and total. So this is the file I'll use for my data set. Just select that, and I'll just call this mean data. And we have some options here regarding whether it's going to be a wide file or a tall file. This was a wide file because it has one column per species. So that's how this is configured. And now I'll create a few tasks. So we're talking now about this TMD model. And so the tasks here pertain to different experimental conditions, in this case, doses. So let's create one task, which would be the TMDD model. Let me go ahead and select the model file. That's right here. It's an SP project file. This is what you've created in SimBiology. And once we've loaded this in, you see that it populated the available models within the SP project, and TMDD was the default. And we see, as Iraj mentioned in the slides, a number of fields being populated automatically. All the variants that are available in this model file, the doses, the species, etc., are all pre-populated into these tables. And to create the experimental conditions, we have to specify which of these are to be included. So we are considering the case where the in vitro affinity was 0.4. So that's this first variant. And this is a SimBiology variant. We'll include that by clicking the arrow button here. So you see the ones which are included on the right and the available ones on the left. And now for the first experimental condition, we would like to have a simulation at a dose of 3 mg per kg. So I'll include this dose. And you can also include multiple doses if you'd like to stack them, but in this case, we'll just use one. And the model, when we run this simulation, it will simulate everything in the model, but you don't have to save all that output if you don't care about it. So here you have the option of choosing which species to include for saving. And that's useful for reducing the footprint of the data files which are produced. So in this simulation, we care about the concentration in micrograms per mil, the target levels in nanograms per mil, and the target levels in the total target levels in nanograms per mil. And you can also multi-select, as I just did here. And again, we have the option of turning off rules if we think that we would like to experiment with that rule being deactivated. Likewise for reactions. In this case, we don't need to do that, but we have the possibility. We need to specify what are the time points that we care about. And so here we're going to be simulating the first 21 days. And this is just a MATLAB vector expression. So we're going to say 0 to 21 increments of 0.1 days. That specifies the output times. This option is if you want the simulation to automatically halt after a particular amount of time has elapsed. That's if, for instance, it's a very long simulation, or it could also be used to prevent sort of infinite loops in the case of, for instance, a failure to meet integration tolerance. So this is sort of a fail-safe in case you don't want it to run forever. This option is to run to steady state. And that's actually a really useful feature, which allows you to specify the model however you like, but the initial conditions of your model file don't necessarily have to be steady state in your SimBiology file. But then if you enable this feature, it'll run the model for some length of time, which you specify here, and it'll keep the final state of the model as the initial condition for a second run with doses applied. So this way you can guarantee that the simulation starts from the steady state, which is useful under some scenarios. But in this case, we don't care about that, so I'll turn this off. Okay, and then let me just name this. This was scenario A, referring to a variant A, and at a dose of 3 mg/kg. And we had 2 doses we consider in this example. So the first was 3 mg/kg. The second one is 0.3 mg/kg, but otherwise they're identical. So instead of having to reconfigure everything a second time, I'll just right-click on this existing task and duplicate it. And now when I go to edit this, everything's as it was for the first task, but now I will choose the 0.3 mg/kg dose and remove the 3 mg/kg dose and just rename this. And now it's configured. So that's a nice time-saving feature. Okay, so we've defined these 2 tasks. We've defined our data set. Let's run a simulation and see how our model compares with the data with these tasks I just defined. So I just right-clicked on the simulation to produce a new simulation, same as for the tasks. And again, there's some descriptive information here. If you click on the pencil icon, you can edit it. And so this is the edit view for tasks, for simulation, sorry. And when you run a simulation, you can specify a number of things. You specify the path where the simulation results should be stored. So every time you run this, you'll get a .mat file for each of the different objects. And so if you want to refer to them later, they're in that directory. The default is sim results, so I'll leave it like that. Data set, if you want to specify a data set, you can use that to compare against data. It's not necessary. You also have to specify the group column if you're using a data set. And most importantly, you have to specify what are the things that you want to run. And so as Iraj mentioned, every item is a combination of a task, which is an experiment, and a collection of virtual subjects. And since I haven't defined any virtual subjects, the only option available currently is the model default, which is the parameter values in the model file with the variance applied. So this is our setup. We have 2 tasks using defaults, storing the results in this directory. I think that looks good. So I just hit OK. And now that we've defined a functionality, we can actually run the simulation by clicking on this run button. And so this will launch this wait bar. The first time you run it, it takes a little bit longer because behind the scenes, it's actually building the model, it's compiling it so that it's much faster for subsequent execution. So it takes a few seconds longer for it to build the model first. We see that we finished the first one already, the first task. Now we're on to the second task, still simulating. Rather building. Okay, it finished, and you see that the summary contains now the path of those files. So if you wanted to, you could go and dig them up and see what the results were directly in those .mat files. But instead of that, we're going to go ahead and visualize the results by clicking on this 4th button, which is the visualize button. And that's available for functionalities which perform visualization. Okay, so this is the visualization view for simulations. And so what you're seeing is one big axis, but actually we can specify the layout that's appropriate for our simulation. So we have several options here. I will choose a 2x2 because we have 3 items we'd like to display. So now we have a grid of 2x2 grid axes, and you can specify what simulation species you would like to display on which of these axes, and that mapping is in this table here. So let's put the unbound antibody concentration onto the first axis by choosing plot 1. And besides the plot number, you also have the style option, which is the how the line should be displayed. So you can have solid lines or dashed lines, etc. And let me just go ahead and map it for the second and third options as well. Okay, now you see that nothing's being displayed because I haven't selected anything yet. So here you have the simulation items that we defined, and you can choose what the name of that item should be in the legend, what the color should be if you choose to set the color, and whether or not to display that. So if you click this include button, it'll display that on the axes. And so by choosing both, we're now comparing the first task and the second task in blue and red on each of these different axes, so each of these different species. And we can also configure, as I said, the line styles and so on. So everything is being shown here. Okay, so this is nice. This is what the model does if you just include the default model parameters. But we want to see how that compares with data. So these second 2 tables are controlling the data to display. And the the data are coming from that file. They have different group numbers assigned to them, and it's up to you to know which group is which. I know that group 1 is the data collected for 3 mg per kg, so it's in the same color as the simulation, so that's convenient. And if you click on this button here, it'll include it, but we still have to map it to the different plots as well. So here we have the data names, which are analogous to the simulation names. So we know that PK3 is the equivalent of the unbound antibody concentration. And so I'll put that on the same axis, which is axis 1. Okay, so now that I've mapped the axis number and I've included it, we see the data are being rendered on the axis. And so we see right away that the simulations don't exactly capture the data because we haven't even calibrated it. So that's perfectly fine. And I'll include also the other data set to include. So that's the free target on axis 2 and the total target on axis 3. Okay, so things are quite off when you use the model defaults, but that's fine. Okay, so the next thing to do then would be to run an optimization to actually improve the agreement of the simulations with the data sets. So if we want to run an optimization, we need the tasks defined. We have that already, but we also need to define the parameters we want to use for optimization. So to do that, we again go to the Explorer on the left, and we choose parameters, and we add a new parameter set. And here, all you really have to do is just select the file which contains the parameters. So that's already here in this directory. It's called parameters. And as Iraj mentioned, if you peek inside, you see that it's just a list. It's an Excel file, and it just contains the names of the parameters, the scales used, whether it's log or linear, and the ranges and an initial estimate for the parameter values, and whether or not to include those in the optimization. So that's already configured appropriately for this task. Specifying the parameters which should be optimized. Okay, so I'm just going to call that new parameters. It's fine. Okay, so now we have parameters defined. We have tasks defined. We can actually start to work with an optimization. Okay, so let's make a new optimization. Here it is. And when you go to edit it, you'll see all the parameters available for optimization. Now, a lot of this we've mentioned already. Firstly, you can choose which algorithm to use for optimization. You have a couple options by default. Those can easily be expanded, but for now, it's depending on the availability of the toolboxes you have installed, which is, if you have the global or local optimization toolboxes. I will choose particle swarm optimization, which is a kind of a global optimization, and the parameters that we just defined. And when I selected this, you see that this table populated with the value, the contents of that file, just for your reference. It's not something you can modify in this view. We choose the data set we want to optimize against. That's one we defined previously. The group information, the ID information. And here we have to define what we want to optimize. So we want to match this task, which we had defined earlier, against the data from group 1, and this second task against the data from group 2. So this is where you define how the simulations relate to the data sets you have available. And likewise, you have to define how the species in the model relate to the species in the data, because they're not named exactly the same way. So here I'm just mapping the total target with the total target, the free target with the free target, and the antibody concentration with the PK3. So again, it's up to you to know how data correspond to simulations. You do have some additional freedom here. If you wanted to, for instance, transform the data because you know that they are normalized relative. For instance, if your data set contains percent change from baseline, but your simulations are just the values, then you could actually transform those simulations with this algebraic expression by, for instance, dividing by the first value, like x1, and that would give you some transformation of the simulation, which is then directly comparable with the data. So that gives you additional degree of freedom for mapping data and simulation. Okay, you have some other options here. For instance, you can fix the random seeds. You can change the directory where results are stored. You can use initial conditions from the data set directly if you want to. I won't do any of that. I'll just use the default value for the path. And now that this is defined, you can run it. I will actually skip the running just for now. Instead, go directly to the visualization view because that has some interesting features. So again, we have grid layout. We have to map the axes. We can map the PK, the free target, the total target. and it's still nothing is showing up because I haven't configured it totally. We choose which options to include, and now you see the data shows up when you do that, but the simulations are not there because we haven't specified what the parameter values should be. In fact, we haven't run any optimization yet. But what you can do is you can start adding profiles. So if you click here on this plus button, you add a profile, and you choose the source. And this source, the only one available now is the values contained in that parameter file that we specified. So I do that, and when you specify the source for this profile, you see this table populates with the values in that file. And if you hit apply, it'll simulate with those values, and it'll plot the result. So now we see, okay, this is the result you get if you just use the defaults in that file. But this is actually a very powerful interface because this allows you to choose the profile, and you can manipulate it directly in this view. So for instance, if I duplicate this with this button here, I have now two of the same. And in the second one, I will, for instance, adjust the V1 parameter value. So you can do that directly in this table by just typing in a new value. To me, it looks like V1 is too large, so I'm going to decrease this. And now I've got a new value. I still have to hit apply. And so it's going to run with the new value. And you see that now we have two sets of lines on each graph. That's because we have one was the original and one is the modified. And you can always tell which is which because if you click on it in the graph, it'll automatically update the number here telling you what it is and the values in the table. So this is actually fully interactive. And likewise, if you select the value in this table here, it'll navigate in the graph. So this is great. This allows you to directly manipulate and compare and turn it on and off. So this is a really powerful way of exploring your model parameters. Of course, on top of that, you can actually run the optimization. We're a little bit short on time. Let me just run it for 30 seconds or so, so that we get a sense of what this is doing. So I specified particle swarm, and so this is launching the this is the built-in MATLAB optimization plot updates. This tells you what the optimization is doing as it's running. And if you're not familiar, these little dots make it hard to see. These little dots show you the current value of the objective function. But as it goes, that value is dropping because it's optimizing, it's minimizing the error. And so this will stochastically converge to a global optimum. Don't have a lot of time, so I think I'll abort this just because we're gonna run out of time here. Okay, so I hit stop. That will, it may take a few seconds. Yeah, there it goes. It stopped, and it's using the intermittent value, which is not really an optimum. But so as soon as I stopped it, or as soon as it completes by itself, it'll produce a new virtual population with the values in that optimization. And so this is the name of the Excel file containing that value. You see also that in the virtual subjects that appeared automatically, so now it's available for use in simulation. And if you go to visualize, back to this same view I showed a moment ago, I can add a new profile, which then uses the optimized parameter values versus the initial parameter values. And if I hit apply, it'll run that. And actually, it's looking already a lot better, right? You see that that the new one is agreeing much better with the data than the previous one. So clearly, it did optimize somewhat, although I didn't let it run to completion. Okay. So this will be kind of the starting point for your cohort generation. For instance, if you wanted to, you can actually export this directly as a new parameter value set. And so this will allow you to jump right into the cohort generation. I'm just going to use the initial cohort generation. Okay, so I want to at least get this cohort generation in there before I run out of time. Let's define the acceptance criteria necessary for this. You do it the same way through right-clicking. This is just a file. So I can go ahead and select that value here, acceptance criteria. Again, it's just an Excel sheet with the upper and lower bounds for every species and time point. So let's specify that, and let's create the cohort generation. And I'll just show you how this works. So you jump in, you specify the parameter values, you specify the acceptance criteria file, the group column, all those are the regular setup. Here you can specify how many you want to simulate maximally before you give up, in case it's a very difficult population to produce. You also specify how many you actually want to have in this population. So let's say 100. And you have the option of either saving all of the simulations or just the ones which are valid. I'll choose to save all. You also can specify how you want to generate the population in terms of random distributions. Okay, and then you have to also specify the items. So same as before, the 3 mg per kg dose for group 1, the lower dose for group 2, and the same species mappings as before. The total target with total target, and the PK3 with PK3, and so on. Okay, so that's all set up. It's very similar to optimization, so I don't need to spend too much time here. Okay, so that's set up. You also can specify initial conditions if you like, so that it would make sure that it's valid with multiple initial conditions. Okay, and now when you run it, you see what it's doing is it's running many samples. Many of them are failing because they're not valid. They don't satisfy the acceptance criteria, but it is producing valid samples. And here we have already 25 valid samples. So I'll just let this run until, well, maybe I'll stop it a little bit early just in the interest of time. If you hit cancel, you still have the option of saving the results. Okay, so I'm saving those results. So now I produced some virtual patients, but not the full 100 that I asked for. And if you go to visualize, it's going to run them again because it didn't save them during the generation process, but it'll save them this time, so you don't have to keep running it. But here it's just going to resimulate the ones that I just produced. And so this just takes a few seconds. Okay, so it's done running those. We still have to set up the plotting, so I'll do that in the same way. Concentration here, target there. And here we also want to show which ones to include. Okay, now this is kind of a big mess because you have all these lines everywhere, but that's because every one of these gray lines is an invalid patient. These are all the simulations that are run. Most of them are not valid. So you can turn those off by turning off the invalid, clicking this button here. And that looks a lot nicer. And so this is a view that you'll see very often in this program, in this app. This is a, we call it a shaded error bar plot. So what it's showing is some measure of the mean or the median in this thick line, and then the upper and lower intervals are some values that you specify. So by default, it's the 95% interval. And all that is configurable. In fact, there's an extended plot settings right here. If you go into this, you can really change everything about these plots. You can change the font sizes, you can change the names of the axes, you can change the ranges to plot for that shaded error bar plot. So this is all configurable. And one other nice thing about these plots is that you actually can save them directly to .fig files for MATLAB or PNGs, and that way everything can be directly exported for presentation. So this is a really nice feature that you can just set it up aesthetically exactly how you like it with the font sizes and so on. You can change also the log linear and so on, and everything can be exported directly then exactly as it appears here, along with the customized names for legend items. So that's great. Now, when it comes to the cohort generation, what we see is for every task, we see the simulations that are run, the intervals of those simulations, so all the density of the produced simulations, and these stars represent the upper bounds and lower bounds that we specified. So here you see that every one of these simulations, the valid simulations in the cohort, satisfy the intervals that we had conditioned them on. So this is working correctly, and we can use this cohort and compare it with the initial data if we go back and create a new simulation. You can also change, for instance, the way this is displayed. If you want to show the median or the mean, whether you want to show the traces or just the statistics. So you can change all these features. Let me just go back one time to the simulation, and I'll just show how this VPOP compares with the initial values, and then I'll wrap up. So this is the simulation we had defined earlier. And here we had used the model defaults. Now I'm going to add in some new items, which is the same tasks, but now I'll use the cohort that we just produced. And I'll run this again. And so this is just going to rerun those populations. And we'll see how the cohort compares with the data set that we had. And I guess while this is running, we can already get set up with the questions and answers if there's any. This is mostly what I wanted to demonstrate today. I hope that you get a sense of the features, and especially the fact that that this whole workflow is then it'll be saved in perpetuity. You can go back at any point in time, come back and see exactly how did you define this population, what were the parameters that are used, what are the references, visualize it exactly the way as it was visualized in the paper, for example, if you export these plots directly. So in essence, this is a fully reproducible, portable application for QSP workflows. Let me just show you the result of this comparison. You see, for instance, the initial simulations were very off, but now the cohort is very similar. So I think the yellow and the blue go together, and the purple and the red go together. So this worked kind of as intended. Okay, so let me wrap up there, and we'll take any questions that you may have. Thank you.
Thank you, Iraj and Justin. We do have some questions, and the first one is, have you tried having non-modelers or people who did not develop the model use this app to run simulations? And is it easy to use for somebody who has not done the SimBiology model?
Yeah, so I'll answer this question. So the aim for this model, essentially, the aim for this app is for the expert modelers to use it to develop the models and sort of like be able to sort of like be familiar with every aspect of it, and sort of like design the sessions and all the experiments and all the sort of like simulations and everything. Once they have set that up, like if you have set up the simulations and you have developed the virtual cohort or virtual populations and the scenarios that you want to simulate, then it's easy for non-modelers to do that. But we highly recommend that it should be in collaborations with the modeler, and the modeler needs to set up the scenarios to be simulated and then provide that session file to the reviewer, to non-modelers to play with it. We think that if you don't have the familiarity with the model, you shouldn't design things, but you can simulate things if you want to.
Do you have any recommendations on when to use the different optimization algorithms, and have you done any benchmarking of them?
I think
so go ahead, Justin.
Yeah, so I prefer obviously the global optimizations. I think typically these models are highly nonlinear, and there exist many multiple local maxima and so on. So the local optimization is almost never the correct option. That leaves the PSO, the particle swarm, and the scatter search. I think they're very similar. I think the results produced are very similar. I think PSO performs slightly faster, but otherwise they're quite similar. Iraj?
Yeah, and I agree with that. We've done systematic, essentially, analysis of benchmarking a number of optimization methods, and the results from our analysis essentially has shown that PSO and scatter search across the board and across essentially different QSP models with the number of parameters, they outperform other methods. So our default recommendation would be to use either PSO or a scatter search.
Is there an ability to use this app to run simulations on a cluster, or will this have to run on a standalone computer?
Yes, I mentioned this at the beginning. You do have the parallel cluster option. So it uses the MATLAB cluster interface, like par cluster, for example. So as long as you have the cluster specified and set up within MATLAB, those clusters which are configured are available to the app, and all the parallel functionality will be pushed to the cluster and run there, and the results will return to the local machine. You can also run it on the local machine using this cluster setup. That tends to be a bit slower because of the additional overhead involved, unless you're running a large simulation.
Is it possible to simultaneously generate virtual populations for different interventions, such as different drugs?
Yeah,
yes.
Well, I guess well, I can speak to this. Yes, so if your VPOP generation, your core generation is set up with multiple experiments at the same time, then any parameter set which is produced has to be consistent for all those simultaneously. So assuming that the same parameters pertain to the different drugs, then yes, every parameter value which is produced as a valid parameter value will be valid for each of the different interventions that you specified in that cohort generation.
Is there a limitation for the complexity of models? So the number of parameters to fit data abundance, et cetera?
There is no limitation in terms of the complexity of SimBiology model. As mentioned at the beginning of the presentation, any model that you build in SimBiology and you are able to simulate it in SimBiology, you can incorporate it here. The models that we've tested, the biggest ones have about like 200-300 parameters with the same number of species, like 200-300 species. But there is no limitation per se that if you have a very large scale model, you wouldn't be able to use this. This is essentially the M files that you develop typically in MATLAB, and this is essentially just providing a wrapper on top of that. So theoretically, anything that you do simulate in SimBiology should be able to load in sessions files here as well.
When defining the optimization task, can you have rules about the parameters? For instance, P1 less than P2?
So I answered this question. The way that we deal with scenarios like this, like for example, if you want to have P1 being less than 2, we define this in SimBiology model. So you have P1, and then P2 is essentially, for example, say you have P2, and then P1 is a fraction of P1, and that fraction takes values between 0 and 1. So instead of estimating P1 and P2, we will estimate P2 and that fraction, and then you provide the lower bound and upper bound of 0 and 1 for your flag or fraction. So that would provide the condition that you want. So essentially, the scenarios that you have in mind when parameters are dependent on each other, you need to take care of those things in the SimBiology model, and then just provide the lower bound and upper bound in the parameter file in the Excel format that we have here.
And I think we have time for just one last question. Compared to the SimBiology model analyzer, what are the extra capabilities of the gQSPSim?
I'll attempt to answer this, and Justin, please feel free to chime in. So what we had in mind here is essentially providing the flexibility to define things separately. So for example, here you have tasks which are equivalent of experimental conditions and virtual populations. I think those two things are combined in scenarios in SimBiology, and sort of like the one thing that we do have here is when you are building your optimizations, you can simultaneously fit to multiple sets of data. You can define your tasks with different variants, and then be able to sort of like calculate your objective function. When you are building with working with virtual cohort generations, essentially here we are providing the capability to define your acceptance criteria, which is essentially the range of your measurements, and generate those virtual cohort generations. Again, the idea is to provide capabilities on top of what MATLAB has. Those features such as virtual cohort generation and virtual population generation, currently at least, we don't have them in the SimBiology desktop version.
Great. We do have some additional questions. We will forward all of the additional questions through to the speakers to answer if they can. But we are out of time, so I would like to thank our speakers for today. Iraj and Justin, thank you so much for a very interesting webinar. And I think it's been fascinating, and I have learned a lot. This looks like a very interesting app. So thank you very much for presenting it.
Great, thank you.
Thank you, Rebecca.
Have a great day, everybody, and stay healthy and safe, and hope to see you at a future webinar.